In this section, we're going to begin discussing the fungi. Fungi are heterotrophic eukaryotes. What that means is that fungi do not make their own food. Fungi consume other organisms, decaying material, etc. They can be saprobes, which means, again, they consume decaying material. They can be symbionts, where, they live in the host and provide some host benefit. They can be commensals, where they live in the host and just don't hurt the host, and they can also be parasites. In humans, we often see fungi causing opportunistic infections. The majority of fungi can't make a healthy person sick. And what we have in this table here are different invasive fungal infections and how frequently they occur, and the mortality rate in infected populations. And you can see that, some of these fungal infections have really high mortality rates, and some of them have very, wide ranges of mortality. So what we'll talk about when we talk about the different fungal infections is the different factors that make people susceptible, and why we might see these varying mortality rates. We are not going to talk about all of these different fungi. I have selected the fungal infections that would be most common in the US. So again, these are our first eukaryotic infections that we've talked about. Remember, bacteria are prokaryotes, viruses are not cells at all, and then we have the fungi that are eukaryotes. And so what's really important for you to think about when you're thinking about clinical medicine and thinking about fungal infections is how does a fungus, being a eukaryotic organism, complicate treatment? When we're talking about fungal infections, particularly in people, we can have yeast form, which is like a unicellular form of the fungus. In nature, they can often exist as molds. Sometimes in people, we will see like multicellular structures. You'll see hyphal structures. Fungi look different often in a human when compared to like a bacterial organism. So using microscopy, you can tell the difference, and sometimes you can even see the fungal structures macroscopically in an infected patient. So yeasts are unicellular. For the most part, they replicate by budding or by fission. So fission is very similar to how a bacterium would replicate, where it would basically duplicate everything on the inside and then split into two cells. Budding is where like a new organism will bubble off basically the original cell. Molds are usually multicellular, and they form these structures called hyphal structures that make up a mat of like interwoven fungal. The hyphae kind of almost look like fibers. So you've got these fibers that weave together and form a structure called a mycelium, or like a mat of fungal structure. If you're interested in fungi, Dr. Mujic teaches a fungal biology course. He is a mycologist, or a scientist who studies fungi. He's not necessarily a medical mycologist, but he knows a lot about fungi, and definitely, if you find fungi interesting, you should take his class. Fungi can reproduce by spores, and they can reproduce sexually or asexually. Remember, bacteria don't have sexes. Bacteria are just bacteria, so they only can reproduce asexually. The spores formed by fungi are reproductive structures. That is different from the endospores that are formed by bacteria. So I would refresh myself on endospores in bacteria so that you don't get them confused with reproductive spores of fungi. And some fungi can be dimorphic, meaning they'll switch from a yeast to a mold. Here, again, you have kind of that nuclear fission, you have budding, and you can have the formation of these kind of multicellular structures. And so this is what hyphae look like. They're these filaments or fibers. They can be septate, meaning there's basically a cell wall between each nucleus, or they can have no cell wall, where it just looks like one giant cell with multiple nuclei. There are a lot of different fungal structures. We're not going to talk about all of them simply because we don't have time, and they don't all cause human disease. Again, if you find fungi interesting, like if you find this unit really interesting, I highly recommend taking Dr. Moyich's fungal biology class. When we talk about fungal infections, a fungal infection is a mycosis. And there are a number of different types. We will talk a little bit about each of them. There are superficial that are limited to the superficial surfaces of your skin and hair. These are mostly cosmetic. You typically don't have any sort of inflammation, no itching. The only thing you might see is maybe like discoloration or, destruction of like the hair, which again is not painful. There are cutaneous infections that then can start to get into the keratinized layer of skin, hair, and nails. These, you may have a host response. There might be some itching, peeling of the skin. Again, the hair can break, and you could also have discolored nails. Subcutaneous is when we get into some of the connective tissue, maybe the muscle. You start to have abscesses and ulcers, but these subcutaneous infections tend to stay localized. So if you get, an infection in your arm, it tends to stay in the arm and not really spread beyond that. The ones that we worry about the most in our healthy patients are what are called the endemic or systemic mycoses, because these are true pathogens that can make a healthy host sick. One characteristic that all of these systemic infections have in common is that they begin in the lungs. They're all inhaled, and then they can disseminate from there. In the US, we have 3 different endemic fungal infections, endemic mycoses. We have one here on the West Coast, and there are 2 the farther east you go. What we also worry about in some of our patients, especially if you are working in a clinical setting, if you're working in the hospital, you're going to be worried about opportunistic fungal infections. So these are patients who were otherwise healthy, but they were not otherwise healthy. They are in the hospital, or they are sick, or maybe they have, some sort of respiratory condition like COPD. Maybe they have a catheter that's been inserted. So they are somehow compromised, and then they can get a fungal infection. So these are your patients who were not healthy before. They were in the hospital, they were sick already, and then they got a fungal infection on top of it. And these are going to be our most common type of fungal infections that you might see in a clinical setting. When we talk about fungal pathogenesis, we talk about primary and secondary pathogens. Our primary pathogens are the ones that are the endemic or systemic fungi that can cause infection in our healthy individuals. There are very, very few species that are able to do this. And again, what they tend to have in common is that they are inhaled, and they have virulence factors that allow them to breach the defenses of the lungs. They, again, have in common, they have 2 phases of growth. They have a saprobic phase in the soil where they'll form kind of those multicellular hyphal structures like a mold. And then they have a parasitic phase where the mold will form spores. The spores will then disperse into the air, be inhaled, and then that phase reproduces asexually in the host. So by either by fission or by budding, depending on the fungus. There are a lot of different virulence factors that fungi can have. Of course, as we talk about the various fungi, we will talk about their various virulence factors, but the dimorphism can help them hide from the immune system because the immune system has to recognize both forms. They can reproduce at different temperatures. They adhere very well to host tissues and host cells. They are sometimes resistant to phagocytosis. There are a lot of different immune evasion mechanisms. So fungi can be very good pathogens. Again, you're probably not going to see fungal infections as commonly as you might see other types of infections like bacterial or viral, but it is possible to see fungal infections, and these are considered sometimes to be emerging infectious diseases.